Okay, thanks very much. Can everyone hear me? Yeah, okay, great. So Christina's talk actually sets up my talk extremely well because now I'm going to tell you about all of the gory details about how these little guys, these little bacterial cells, and a few other types of cells swim around. So the focus of my work is basically on the mechanics, the biomechanics of how single cells swim. And today what I'm going to tell you about, amongst other things, in addition to an overview of some other types of cells, is how these little marine bacteria, which are only about a micron or so in size, can use this whip-like flagellum that you saw on a couple of Christina's images, how they can use this flagellum to actually turn. And it might sound mundane, but I'm going to try to make this very exciting, and I think I'll be able to show you that not only do these things have some really interesting material properties, but they can inform us on how to think about things like designing robots and micro-robots. And it's actually quite an amazing little structure and process that this thing uses. So as Christina also mentioned, I don't expect you to know any of these people. The reason why I put them up here is to really show you the diversity of the people that are working in this field. So these are some of my collaborators on some of the projects that I'll talk to you about. And what I just want to point out is we have people that are environmental engineers, mechanical engineers, physicists, biologists, mathematicians, ecologists, applied mathematicians, bioinformaticists, and a whole range of different people working on these problems together. And even my own background, I have undergraduate degrees in physics. I've done my PhD in engineering. I've done postdocs in physics. I've done postdocs in environmental engineering, and now I'm working in a mechanical engineering department. So we're all speaking the same language of trying to quantify how these little cells move around and how their interactions add up to these things like flocking and swarming, and trying to describe nature and maybe understand something about how we can engineer some microsystems using these small little swimming cells. So here's another cross-section of some microorganisms that you might find in the ocean. Now, these aren't all single cells, and they don't all swim, but the one thing that they have in common is that they're excellent engineers. And the reason why I say that is because you have some small cells that are experts at nanoengineering, right? So they can make these calcified plates that are terribly intricate that armor the cell. There are other cells that have propulsion systems like flagella. There are cells that have many flagella that cover them, like this paramecium, that can synchronize all of these little hairs to beat in unison on the surface of the cell to propel it, and a whole other array of different morphologies and shapes. But what I'm going to focus on today is how single cells move. Okay, and you've seen a few of these pictures from the previous talk. These are the two things that I'm going to focus on. One is this Chlamydomonas cell. This is what's called a biflagellate, so it has two flagella, and they're bendy flagella. And these are bacterial cells that you saw have this corkscrew-like flagellum. And the one thing that I want to point out before I get on to sort of the science is some of the methods that we use. So someone asked about microscopes and how we quantify these things. So of course they're very small, right? So these are microns in size. So we have to use very high-powered optical microscopes to capture these things. The other thing that I want to point out is how fast these things are. Now, the speeds might not be very large, but the frequency at which they beat is very large. So this is slowed down a lot. This is slowed down probably about 100 times. So this cell is actually beating with a frequency of 50 hertz. That means in 1 second, this thing has beaten its flagella 50 times. When you look into a microscope, it's just a blur. So one of the techniques that we have to use are some specialized high-speed cameras that can take pictures at 500 to 1,000 to even up to 10,000 frames per second. To put that in perspective, the camera that you have on your cell phone or a YouTube video is about 30 frames per second. Okay, so it might look very slow and very easy to quantify, but there's some tricks to it. Okay, so why do we care about single cells? Well, they're important at all scales, really. And this is about the largest scale that we know of for living things, our Earth, right? We don't know of anything else living on any other planet, so this is the biggest picture I could find. And all of this green stuff that you see out in the ocean is actually the source of a lot of the oxygen that we breathe. And it's not from things that you might think of, like large kelp forests that we might have off of the coast here in Santa Barbara. But actually, all of this green stuff is single cells that float around in the ocean and are photosynthetic. Right? So these single cells are having a global impact. Now, they're not motile, but I just wanted to show this for effect. What is motile and does have very large-scale features are what are called red tides. So you might have seen these or experienced them perhaps where you live. And these are blooms of cells that reproduce very rapidly. And when they do that, they consume all of the oxygen, and they can devastate ecosystems. And what's really going on with both these cells and these single cells is the way that they reproduce and the way that they talk to each other is really governed at the single cell level. Right? So the way that they talk to each other is through giving off chemicals, and they can smell the chemicals, basically, that the other cells give off. So that's how they coordinate. That's how they find food. And they also can communicate through physical forces, communicated through the fluid. So I'll talk a little bit about fluid disturbances and what that means for the interactions of different cells. They're also important, of course, in the human body. So sperm cells are sort of the obvious thing. So sperm cells can swim to transport their genetic cargo to egg cells for fertilization. We also have flagella like this that are sort of attached to the linings of lots of our organs, like our lungs and our kidneys. And these beat in unison to move fluid around in our organs, and they perform very important functions. And when they don't function right, these are what are called ciliopathies. So it's a series of diseases that we can get due to the malfunction of these flagella. And then sometimes we have swimmers in our body that we may or may not want there. Maybe I shouldn't have shown this before lunch, but anyway, here it goes. So we have bacteria living all over us. Sometimes you get an infection. That's a bad thing, and you have to take antibiotics to get rid of it. So those are cells that you don't want in your body. We also have good cells living in our body, so cells that are living in our intestines and our guts that do helpful things for us, right? So it's important to understand both types, right, infections and the good types of bacteria. And then on the engineering side of things, we often look to these small-scale engineers to try to basically steal their great ideas. So these little cells can actually, they make motors and they make structures that we still can't even replicate. Even at the best engineering universities in the country, we cannot replicate these things. So we study them, we try to copy them, and we try to learn from them when we design our own systems. So this little guy, this is actually not a living thing. This is actually an engineered microswimmer. Okay, so what it is it's a tail that's comprised of a bunch of magnetic beads that are stuck together. And then there's an external field that's driving this thing to beat. And by driving it to beat, it actually swims. Right? So this is a step in the right direction. And the goal of a lot of this type of research is to develop micro or nano robots for drug delivery. So you can load this thing up with some specific type of medicine and direct it to swim toward, let's say, a cancer cell to kill it off. Christina also mentioned extracting energy from bacteria. So these gears appear to be spinning on their own. They're actually not. They're sitting in a bath of bacteria, similar to what Christina showed in her talk. So if you look very closely, you can see the kind of swirls and fluctuations. And that's actually what's driving these gears. Right? So we might even be able to extract energy from these things. And there are plenty of other applications. Now, to lay down the ground rules, what governs most of the interactions at this scale, since all of these cells live in wet environments, is fluid forces. So this is the field of fluid mechanics or fluid dynamics. Some of you may or may not be familiar with any of this. It doesn't really matter. I'm just going to give you some very basic concepts. So in fluids, we often think of two different types of forces. There are inertial forces on bodies that are immersed in a fluid. Right? So these are changes in momentum that confer forces. And then there are viscous forces. So this is due to sort of the stickiness of the fluid, right? How much it resists a shearing motion. And we often want to know in which regime a process is. Is it dominated by inertial force or is it dominated by viscous forces? And I'll give you an example of this in a second. So what we do is we take the ratio of these forces for some body that's sitting in a bath, and this ratio is what's called the Reynolds number. It's a very famous number in engineering and also in physics. And if the Reynolds number is very large, that means that inertial forces are important, and momentum basically dominates. If the Reynolds number is very small, that means that viscosity dominates the system. Okay? Basically what this boils down to, this Reynolds number R, is the size of the thing. If it's a cell or a person or a cruise ship, it doesn't matter. The velocity of the thing, how fast it's moving in the fluid, and then rho, which is the density of the fluid, and eta, which is the viscosity. So eta and rho are basically fixed. We're only talking about water, right? So the Reynolds number is basically proportional to how big something is and how fast it is. So if it's big and fast, it's inertial. If it's slow or small, then it's viscous. Okay. So this is an example. So this is what this is a tray of fluid with a little marble in it. And when I start the video, what you're going to see is that the tray is spinning around, and the fluid is very viscous. And that makes this Reynolds number very small. And the thing that I want to point out is that when the tray stops rotating, the marble stops immediately. Right? So you can see how viscous it is. Right? So there's a little tiny bit of motion, but not much. Now this is at Reynolds number 30. So now we're using water instead of this honey or syrup. And now watch when the plate stops. So the fluid keeps turning and carries this little bead around. Right? So why am I mentioning this? Well, the reason why I'm mentioning it is because bacteria can't coast by on their good looks or on momentum. Right? They are unable to coast. If you're swimming in a pool, or if you dive into a pool is an even better example, you can kind of coast through the fluid without kicking your feet. Bacteria can't do that. If they stop moving their arms, they stop moving totally. All right? So that's a very important point. So what are the Reynolds numbers for some typical things? So for a human, it's maybe about 10 to the 4th. Michael Phelps is probably about 10 to the 5 or 10 to the 6. Fish are a little bit smaller, maybe about the same speed, so that's a little bit smaller yet. And then when you get down to the size of bacteria, their Reynolds numbers are about 10 to the minus 4. So this is very, very small, much less than 1. This means that bacteria and microorganisms are totally dominated by viscosity. All right. So with that said, this is another very important notion in hydrodynamics. So when the Reynolds number is very, very small, because there's no coasting, there's a principle called reversibility. And what this gentleman is about to do is he has this little apparatus that's, let me just draw it on the board. So it's an apparatus that's 2 concentric cylinders like this. And the outer one's fixed. There's fluid in the middle. And then what they're going to do is rotate the inner cylinder. And he's going to do that with a little drop of dye. So let's watch the movie and see what happens. All right, so here we go. All right, so he's putting in the dye. And this is something like corn syrup, so it's very, very viscous. And he's going to turn the handle very slowly. So the Reynolds number for this experiment is about 0.01. So it's very small. All right, now he's going to rotate this handle. And you see the dye droplet gets sheared and smeared out into this band. It's kind of hard to see. And he's going to wind the handle around about 4 or 5 times. And now this isn't a Hollywood trick or anything. We're not playing the video in reverse. This gentleman is just going to now unwind the cylinder. And we're going to see what happened to our droplet after he unwinds it. This is very exciting. I know, it's suspenseful. But it's truly amazing. Right? Yeah, it's a beautiful experiment. I didn't do it, but it's it's actually G.I. Taylor. So we actually do this in the fluid mechanics class that we teach. We built one of these devices and we show the students this principle. So you might say, okay, well, what does this have to do with swimming? It actually has a lot to do with swimming, and it's actually comes from this theorem by a guy named Ed Purcell, who came up with what's called the scallop theorem. And Ed Purcell is actually much more famous for something else, at least outside of our circles. And that is that Ed Purcell got the 1952 Nobel Prize for discovering NMR, nuclear magnetic resonance, which is the basis for MRIs, which is a very big deal. But after that, I guess he got sick of all the fame and fortune of working on NMR, and he turned his attention to swimming microbes. And he spent the latter half of his career doing some truly pioneering and amazing work on trying to understand the micro hydrodynamics and transport properties of swimming cells. So what Purcell said is because of this reversibility and the fact that things can't coast, that limits the strategies that cells can use to swim. It's called the scallop theorem because Purcell described the simplest thing that might be able to swim as something that looks like a scallop. So this is my less artistic rendition of Purcell's drawing, but it actually moves. So it's two rigid links, think of these as flagella or clamshells or something like that, connected by a hinge. So if you ask this thing to swim, it'll do the only thing it knows how to do, which is close its shell, and it moves forward, so that's great. But then the problem is it can't coast, and it can't do anything else with its arms besides reset them to make another stroke. The problem is when you reset, you go back to where you started, right? So there's no motion, right? So cells have found different ways to get around this, and there are two main types of flagella that do this. One type is what's called the prokaryotic flagellum. That just means that these flagella are on cells that have no cell nucleus, okay? And these flagella are actually rigid, well, essentially rigid, and they're driven by a rotary motor that's embedded at the base of the flagellum. So this corkscrew just gets spun and basically drills the cell through the water. Right, so you can see that happening in this video from Howard Berg's lab in Harvard. This is a picture of the motor. So this is what it looks like. It's about 100 nanometers across. Here you can see this video from Howard Berg's lab as well, where this is the flagellum, this is the cell membrane, and then you go inside, and here's the motor. And we actually know, not me, but other people that are much smarter than I am, know a lot about these motors from doing scanning electron microscopy of them. And these are absolutely incredible structures. They look, if you're an engineer, exactly like an electric motor that we would build. They have rotors, they have stators, they have bearings, they have all the things that we would put into a motor. And this is what drives these cells by this flux of ions that comes through the turbine, essentially. Okay, so the other type of flagella is a bendy type of flagella. These are found on eukaryotes, which means that they're on cells that do have a cell nucleus. And these are things like sperm cells and this Chlamydomonas cell that we talked about. So sperm propagate bending waves down the flagellum. And again, if you notice the time scale here, this is taken at about 1,000 frames per second. And the beat frequency is about 50 hertz. Okay, so 50 times per second. The way that they do this is the flagellum is actually made of these striated materials. So there are fibers that run all the way down the flagellum. And if you look at a cut through the flagellum, it looks something like this. So these are those fibers that run axially. And there are little tiny motors in here that sit between these filaments. And what those motors do is they walk between the two filaments, and they create a shearing action. And under the right conditions, when you create a shearing action, it actually creates bending. So that's what's happening with these guys. Okay. All right. So now back to the fluid mechanics. So we know from Newton's laws that if the fluid is exerting a force on the cell, then the cell's got to be exerting a force on the fluid. And one of the big questions over the last few years in this field was, what are those forces? And what are the velocity fields, that is the disturbance fields, that these cells create when they swim? So we started off very simply with a little video like this. This is just a little plastic particle. And here comes a cell that's swimming. So this is a Chlamydomonas cell. And you can see that it gives this particle a little kick, even when they're not touching. Right? So it's still moving and vibrating due to the motion of this cell. Right? What we can infer from this is that the cell is disturbing the fluid around it. So what we did was to make thousands and thousands of measurements of little particles like this for many thousands and thousands of cells. Yes, you have a question?
Just from the picture, I would have expected
It's part of the way that the cells swim. So you'll see this on the next slide. It's actually quite cool. So if you do this for many, many particles and many, many cells, you can then make pictures that look something like this. So let me walk you through this. So these little arrows tell you the direction of the fluid flow around this cell. That's basically the direction that those little particles are going. And we've just added up lots and lots of these little particles on this one video. So we have a cell that's swimming like this from left to right. Actually, the key is down here. It tells you where its flagella are. And this is the disturbance field that this cell creates. Now we've mapped out the whole thing. So it looks something like this. Velocity, yeah. So it's the speed of the fluid molecules at different distances away from this cell. And the red lines are basically the lines that particles would trace out if you followed them for long enough. So they go around and around. Now the thing that I want to point out here again is scale, both time and space. This distance is 100 microns. That's less than the size of a human hair, the diameter of a human hair. And the time scale, this is one beat cycle of these flagella. So if they beat at 50 hertz, that means that this whole video lasts just 20 milliseconds. Okay? So that's 1/50th of a second, right? So we actually have to use some very specialized experimental techniques to measure these things. And once we have all of this information, then we can start to apply it to some of these problems that Christina was talking about. So one of these problems is if the cells are in low density, so these are cells in green, and these are more little particles in red. If you watch the cells swim around, what happens is you can see that they disturb the particles as they swim by. And then when another cell swims by, it disturbs them in a different way. So it's just like when you pour cream in your coffee and flick it with a spoon, that's what these cells are doing. They're stirring up the fluid. And if you imagine that these particles are maybe food particles, or maybe they're signaling molecules that these cells use to communicate with one another, this stirring action can be very important for how they communicate or how they find food. And then another limit comes when you start to increase the density of, or the concentration of these cells, so that they're close enough that the flow fields, the velocity fields, start to affect one another. So if one cell is swimming here, the velocity in the fluid that it creates affects its neighbor, and vice versa, right? So as they start to come closer and closer together, these effects start to become more and more. And that's where you get into situations like collective dynamics. So these are high concentrations of bacteria. You can't even see the bacteria. They're so concentrated. And this is just like what Christina showed. So then you start to get these emergent patterns, these whirls and jets. And this is a quantified version of this where the coloration tells you how fast things are moving, so how fast the cells are moving. It's a little hard to see the arrows, but the direction is there too. Right, so you can see that they coordinate over scales that are much, much larger than a single cell. This whole thing is a couple of hundred microns. So a cell is basically a pixel on this display. Right, but the motion is coordinated over much, much larger scales. Okay. So now all of these problems that I've told you about so far have to do with propulsion, with swimming, right, in one direction, let's say, straight. But Christina told you that turning, or tumbling as it's called for bacteria, can also be important. And actually this is a necessary trait that these guys have for survival. So not only do bacteria turn, which I'll tell you about more in a second, but also these Chlamydomonas cells turn. So there's some beautiful work from a group in Cambridge in the UK. And what they showed is that when these cells swim straight, they beat their flagella together. And when they turn, they beat one a little bit faster than the other. And what this is like is rowing a rowboat with one oar, right? So they make sharp turns. Okay? And this is what they use to get away from predators, to find food, to find light, and all kinds of other resources. So even if you only have one flagellum, but it's the bendy type of flagellum, you can also turn, right? So this is a sperm cell that's making a turn, and all it does is bend its flagellum more to one side than the other. That's a little easier said than done because we actually don't understand this process. But you can see that it can do it. We can observe that it can do it. We're still trying to understand it. And this is also very important for what's called chemotaxis for sperm cells. So this is how sperm cells actually find eggs to fertilize. So in the ocean, something like the sea urchin, which you can, I just found out actually there's a nice little aquarium just down the road here that you can just walk into that's near the Marine Science Center. And you can see a bunch of these guys down there. And what these little guys do, these sea urchins, these are about a couple of centimeters across, is they just exude their sperm and their eggs into the ocean. And then they have to meet up. But it's not by chance that they meet up. The sperm cells can actually sense chemicals that the eggs give off. So they can actually smell the eggs, and that's how they navigate to them. So we can actually reproduce this in the lab using microfluidic devices. So it's a little chip like this that's basically a series of little pipes and tubes that's mounted on a glass cover slip or a slide. And what it looks like is something like this. So this is about a millimeter across. And we can inject chemicals that the sperm cells like on this side, and then we can inject sperm cells on this side, and we can watch how they swim toward this chemical. So that's what's happening over here on the left. So here's a sperm cell, and the chemical is over here, but you can't see it. Now you'll notice when the sperm can smell it. So the sperm has this weird behavior where it swims in circles. But then as soon as it smells it, it takes off. And every now and then it checks to make sure it's on the right track. Right? And eventually it gets to the other side. But there's no egg there, so it's too bad for him. Anyway, there's too many jokes that one can make in this field, so we just won't. Okay, right. So that's turning for eukaryotic cells with bendy flagella. All right, now we come to our bacterial cells. So these are the E. coli cells that Christina mentioned about this run and tumble behavior. So I don't have to tell you too much about it, but basically these guys have multiple rigid flagella. And when their motors all rotate in the same direction, they twist up together into a big helix. So you'll see like this super helix. And then when they want to turn, you'll see a cell come in this side. Not that guy, this guy. Right. So when they turn, what they do is they counter-rotate one flagellum, pops out of the bundle, and they turn randomly in a random direction. They can't control where they're turning like the sperm or the Chlamydomonas. So you might ask, well, how in the world can this random turning get me anywhere useful? And it's really not about how you turn or in which direction you turn. It's really about when you turn. Right? So if there's some food over here, and one of these cells is searching for this food, if the cell is swimming away from the food, it can smell that the concentration's getting lower. So it knows that life's getting worse. So it turns more often until it randomly ends up swimming in the right direction. And when it starts swimming up the gradient, it knows that life's getting better because it can smell it. So it just doesn't turn as much. Right? And that's how a random process can actually direct you toward some physical goal. Okay. All right. And this actually works. You can do this in the lab with these are bacteria that like oxygen. So there's oxygen that's coming out of this little air bubble. And if you track enough of these cells, you can see that they all build up around this interface. Right? So they're clustering around the oxygen where the oxygen's high. Now that brings us to the main part of the story. How am I doing on time? 15? Perfect. All right, great. I think we'll get through this. All right. So the problem now is we've talked about eukaryotic cells with multiple flagella, like Chlamydomonas. We've talked about eukaryotic cells with one flagellum, like sperm. We've talked about bacteria with multiple flagella. And now we come to a bacterium with one flagellum. And these are actually very important bacteria. 95% of swimming marine bacteria have one flagellum. These are very important things that participate in biogeochemical cycling, that's fixing carbon and nitrogen in the oceans, and they participate in the food chain and all kinds of other important stuff. They also transmit disease like Vibrio cholera, which transmits cholera, and all kinds of other things. Now the problem is that our biologist friends, who we really like, we like them a lot, but they got one thing wrong with this cell. They told us that it only swims forward and backward, and it doesn't turn. And it does this by spinning its flagellum one way and then reversing it and swimming the other way. Okay. And what a group from Pittsburgh noticed about 3 years ago, physicists by the way, what they noticed by doing some careful tracking of these cells is that not only do they turn, but they turn a lot. And not only do they turn a lot, but they're actually even better at chemotaxis than their E. coli cousins that have many flagella. So the question that came up is, how do these cells do this? And they turn actually in a very peculiar way. So this is the data that was in this original paper from the Pittsburgh group. And let me just walk you through one cycle here. So green means that the cell is swimming forward. That's head in the front, tail in the back, flagellum in the back. So it swims forward, and then it reverses the motion of its flagellum, the rotation, and then it swims backward and retraces its steps. We understand that, no problem there. The interesting part comes when the cell switches from backward swimming to forward swimming. It makes a 90-degree turn, like that. These are what the original group called flicks, so we have to stick with this nomenclature now. And the question is, how do they do this with one flagellum? The reason why it's an interesting question is because these cells only have one motor. And we know this because we know the details of their anatomy very, very well. And all that motor does is spin the flagellum, just like every other bacterium that we know about. But what you can see here is that in this very tantalizing video, the cell is originally, so it starts out backing up right here, and then it looks like its flagellum bends, and then the cell takes off in a different direction. So that's what's shown here, right? So here's a straight cell, and then here it's bent a little bit, and then by the third frame, it's basically swimming off in the new direction, right? And this is with conventional imaging. This is 30 frames per second imaging, just like your cell phone or YouTube, et cetera, et cetera. So this is what really got us interested, and we want to know what the mechanism was that was at play here. So this is what we did to figure it out. We tracked some of these cells at very high speed. So this is at 1,000 frames per second, right, about 30 times faster than the original study. And what we noticed is that, so this is about to do one of these 90-degree turns. Okay? So it starts off backing up. So we know the flagellum's over here, even though you can't see it. And what I want you to notice is what it does right before it turns. So when the track turns red, that means it's swimming forward, and then the turn happens. Okay? So that little portion where the cell swims forward only lasts about 10 to 20 milliseconds. That's less than one frame of video, conventional video. So no one would have seen this otherwise. But this actually gives us a big clue as to how the cell is turning. But we were still a little dissatisfied with this. So what we did was we used a different type of microscopy so we could see the flagellum during this process. Now, it's a little bit slower in terms of the frame rate, but we can get a better picture of what's happening. So here the cell is backing up, and these are exactly the same videos. It's just that here we've marked the flagellum. And what you should notice is where the flagellum bends. Right, so after it switches to forward swimming, you can see that the flagellum bends at the base. So we have these two clues. Right? They're swimming forward, and the flagellum is bending at the base. All right, so what does the fact that they're swimming forward mean? Well, if you do a little free body diagram of the propulsion of these cells, if they're swimming backwards, the flagellum creates the thrust, so that pulls this way. Forces are balanced, so drag is equal and opposite to thrust, so it's got to go that way. So the cell is under tension, right? And then when the cell switches direction and swimming forward, as is the case just before they turn, the thrust is now pushing the cell, and the drag is now pointed back at the thrust. So now it's in compression, right? So we have a long, thin structure like this flagellum that's in compression. And there's also another little bit that we have to consider. This is a little piece that connects the flagellum to the motor, which is down here. This little thing is about 100 nanometers long, and that's what's called the hook. For historical reasons, it's called that. So it's the same diameter as the flagellum, but it's very short. Okay, so we have these two structures, the hook and the flagellum, that are under compression. So when you think about long, thin structures that are under compression, in engineering, we think about buckling. And we think about it a lot because we don't want it to happen. So buckling is when you compress these structures, and they have no place else to go but bend outwards. And that means that structurally they failed. So this happens in train tracks, in bridges, and in beams. And this is what it actually looks like. This is a little experiment. I'm sorry I don't have the citation. I really should have it on here. But what this is a thin beam, and this gentleman's putting a beaker on top, and he's going to fill it with some metal shot. And the characteristics of buckling, there we go, are that it happens very suddenly, right? So this beam works great. He's starting to fill it with shot. It works great. And then all of a sudden, it doesn't work at all. Right? So that's buckling. It's an instability. It happens very suddenly at a critical value of the compressive force. And we know what those what those critical forces are based on the beam itself. So the critical force, the critical compressive force or weight, is pi squared times E times I. That's what we call the rigidity. E is the Young's modulus. That's like the flexibility of the flexibility is a bad term. It's like the spring constant of the material. I is what we call the area moment of inertia. That's got to do with the cross-sectional area of the beam. And then L is just the length of the beam, right? So the stiffness EI, if it's really wide or it's not very springy like steel, then that means it's very stiff and it's resistant to buckling. If the stick is very long, if the beam is very long, that's in the denominator, so that makes this critical force small. So the longer the thing is, the easier it is to buckle. Now, I'm sure you've all experienced this in your daily life. Whenever you've gone to McDonald's and you've grabbed a straw and you can't wait to get a drink after you've chowed down on your chicken McNuggets, you slam the straw on the table and it cracks in half, right? So that's buckling, right? You're compressing this long, thin straw, and all of a sudden it cracks unexpectedly, all right? So that's what we think is going on here, is that one of these two structures, either the hook or the flagellum, is buckling. So we can estimate the compressive load on these cells, which is what we call the hydrodynamic load because it's due to fluid forces. And that turns out to be about a piconewton. And we can also calculate the critical load for the flagellum to buckle. That turns out to be about 50 piconewtons, right? So there's no chance actually that the flagellum is buckling. But then when you look at the hook, it turns out that the hook has an EI, the stiffness, that's about 10,000 times less than the flagellum. So the critical load for the hook is actually about the same as the force that's on it. So we have this chance that this thing is actually buckling under the hydrodynamic forces, right? So then the question is, how do we test this? So just to recap the hypothesis, the cell is doing one of these 90-degree turns. After it backs up, it now swims forward. Forward means that there's a compressive load on this little hook. When the compressive load is too high, that hook buckles, and the cell turns, right? This is what we think is happening. So now we have to prove it. What we can do at the macro scale, since I'm kind of trained as a mechanical engineer, I have a lot of civil engineering friends, what we would usually do is take this thing to the mechanical engineering department, put it under some hydraulic press, and then start squeezing it until it breaks. Okay? But we can't do that, right, because it's a little too small. Right? So we need some other method to do this. And also, we already think that it's already breaking under the loads that it's under. So what we actually need to do is reduce the load and wait to see when these things stop turning, right? So if they stop turning, that means they stop buckling. So we have a nice way of doing this, which is because the load, the hydrodynamic load, is proportional to the speed, if we can reduce the speed of the cells, we can reduce the force, and if the flicking goes away, that is the turning goes away, then that means that they've stopped buckling, right? So the way that we reduce the speed of these cells is actually quite nice. The motors actually run on sodium because these things live in the ocean. So if you take a little bit of salt out of the water, then the cells actually start to slow down. Okay? So at a moderate swimming speed, a pretty high swimming speed, that's this cyan, the cells swim around very happily. You can see that they do these turns just like they did before. There's no problem. But then when we slow them down to about 10 microns per second, something really dramatic happens. Right? If you slow them down, you've taken away the force that buckles the flagellum, or that buckles the hook, and now they just swim back and forth. So what we can do is watch every time they change direction. So either a reversal like this, or a 90-degree maneuver like that, and we can count the fraction of times that they reorient using this type of behavior. And if we plot that fraction as a function of the swimming speed, right? So this is the probability of flicking as a function of the speed of the cell. And remember, speed is basically force, and flicking or turning is basically buckling, right? So what we see here is that as the force increases with the swimming speed, we have this sudden transition between a state where the cells don't turn, right? So this is the likelihood of buckling or turning, to a state where the cells turn all the time, right? And it's really this sudden change in this turning behavior that tells us, quantitatively that this is how these cells are turning, that it's a buckling phenomenon. Okay. So I think that's just about my last slide, and I'll just kind of conclude with a little bit more sort of perspective and some speculation. One of the things that I want you to take away from this is that this is not some parlor trick that some bug in some remote corner of the ocean does. Rather, we think that this is a common thing that these things do. We think it's basically ubiquitous. So the way that we show this is we have lots of graduate students at MIT that can't wait to graduate. So what they do is they volunteer to go to the ocean and get a bottle of seawater. So this is my colleague Kwang Min. He actually just had a baby yesterday. Well, his wife had a baby. What we did after Kwang Min brought the seawater back to the lab was we put it on a microscope, and we looked for some big juicy dead thing like this copepod, right, which becomes food for all of these little bacterial cells. And when you watch these little bacterial cells swimming around, you can track them very carefully with the microscope, and you can see that they all basically exhibit this 90-degree turning behavior, right? So that's a common thing that many of these guys do. We've kind of marked some of these with little markers when they make these turns. And it turns out that something like 60 to 70% of the cells that we look at do this behavior, right? So that's what kind of tells us that it's a pretty ubiquitous thing and it's pretty common. So these bacterial cells have known about this for a long, long time, but we're just now starting to kind of de-engineer them or deconstruct them, if you will. And just as a little perspective on these things, the research that we're doing here is very important for material science. The materials that make up these hook structures and these flagella are just single proteins that tile together into complex structures. And yet they can do these complex tasks, and they can undergo huge strains and huge deformations, which are very hard to achieve with other materials that we can engineer. And it's also very important, as I said early on, for microrobotics. So what this cell is doing is actually something that is highly sought after by roboticists. It's called under-actuated dynamics. And what that means is you get a 2-for-1 deal, right? So you have one actuator, that's the rotational motion of this motor, that gets you 2 degrees of freedom. So one rotational motor allows me to swim. It also allows me to turn, right? So that's a great deal. And the reason why we think they do this, and this is more speculation, I would say, than science on our part, since we're not evolutionary biologists, but one can think about this as a kind of biologically cheap way to achieve this functionality. So what I mean by that is bacterial cells like E. coli that have many flagella and many motors live in our guts. So they have the luxury of lots and lots of food. So they can construct as many motors and as many flagella as they want. But these cells live out in the ocean, which is not like a mixed soup of material. It's more like islands of nutrients that are sparsely distributed. So these guys can have very lean times where they don't have any nutrients. So they have to conserve how they use their resources to build these structures. And that's more speculation than anything. So I'll leave it there and take any questions that you guys might have.
To put the things in perspective, looks like the flocking or the collective behavior has to do with the flagella, right? The velocity field created by them and that collectively move. But what about those bacteria or those cells which do not have that antenna-like flagella? How do they show collective behavior?
Right. So I glossed over this just in the interest of sort of time, but this is actually a hotly debated area right now, which is how important are these fluid forces that affect your neighbors versus steric forces, that is just banging into one another. And there are sort of two schools of people, since we're talking about flocks and schools, there are two schools of people. One group sort of thinks it's the hydrodynamics, one group sort of thinks it's more the steric, but in actuality it's probably somewhere in between, or a transition from one length scale to another. So when you're far separated, it's hydrodynamics, and then when you bang into one another, it's steric. So I think it's probably a combination of both of those things. I'll let the moderator pick the...
So in in a system like a metal bar or a bridge, buckling means damage. And this apparently is not damage. How common is buckling with no damage?
It's actually more common than you might think in biology. So, but this is still the most widespread example that we have really seen. I could be wrong on this, but actually, whenever you see leaves on trees that have ruffled edges, that's actually a buckling instability. It's sort of a static buckling instability. The Venus flytrap that snatches, so it's a plant that snatches bugs, that actually closes its trap by a buckling instability. That's why it's so quick and so fast. When you touch a little hair on the inside of it, it actually triggers this buckling instability and it snaps shut. So there are plenty of other examples, but they're kind of isolated and very specialized. So this is actually a big thing that a guy at a colleague of mine from MIT works on. And what he calls it is functional failure. Right? So you can actually design into systems the ability to undergo large deformations but still recover. Right? So this is actually something we can do. We've just never tried it as engineers. Right? So good question.
I'll just make a comment. Biologists tell me that flagella do break occasionally. I don't know under what circumstances.
So what, bacterial flagella or? Yeah. So bacterial flagella, the most widely studied flagella are actually those of E. coli, which undergo, this is a bit esoteric, but they undergo these what are called polymorphic transformations, which are not actually buckling or breaking. I don't actually know how you would classify it, but it's something a little different. So yeah, they do bend if you watch these videos of I don't know if I can get back to it in a reasonable time, but probably too far back. But you can actually see that the E. coli flagella are quite bendy. Yeah. So if you watch these things, when they decouple from the bundle, they actually can like bend in half and things like that. So it's bending, but it's not buckling, I don't think. Yeah.
Yeah, I'm wondering if your buckling and effect the locomotion is pretty much like pH dependent, like the more, the higher the hydrogen ion concentration, it seems the more activity you get. And, you think of maybe fertility studies, which is sort of to some degree pH connected, and also, basically, what is it that they're sensing? Is it really just a local stream change in the pH for, say, the sea urchin sperm to find the egg? I'm sort of wondering, something is driving that. Is it an electronic attraction? Yeah. What are we talking about?
Okay, so there's two questions here. Let me tackle the second one first. So the second one is, how do the sperm cells detect the egg cells? What it is the eggs are actually doped very heavily with peptides. And the sperm can sense very specific peptides that only an egg of its species emits. So they have the sperm cell has sensors on it that when a molecule of this peptide bonds to this sensor on the cell, it then sends a signal inside the cell to tell it what to do. Right? So it's very... Sorry? It's actually calcium signaling that's transduced through GCMP, I think. I forget what the molecule is or something like that. But it's very specific. And the reason for that is because you don't want cross-fertilization of species. So even closely related sea urchins, for example, the two different species will actually use two different peptides to attract their own sperm, right? So that you don't get cross-fertilization, which kind of dilutes the genetic material. Okay. And then the first question was. Yeah. Yeah. No, that's a good question. So with the sodium modulation of the cell speed, this is a well-studied thing by biologists, and actually we maintain the pH at one level, even though we're changing the sodium concentration. So we buffer the solution. And it's pretty well known by our biologist friends who, well, we take their word for it sometimes, but they tell us that all this really does is limiting the number of sodium ions that can go through this motor really just slows it down and has no other physiological effects or biochemical effects on the cell. So, yeah.
Right there?
In the back, yeah.
So the success of each buckling seems to be random, and then the bacteria takes a few more strokes to see if things improved?
Yes. Yes. So it works on an algorithm much like these guys, except with a different way of turning. Is that the question?
Yeah. So but it doesn't really know which way it's going to wind up after the buckle.
No, that's right. Yeah, it's still random, just like this is random with the E. coli.
So if things improve, it just says, okay, keep swimming that way.
That's right. Yeah, yeah. So they modulate the time in between when they change direction, so that changes the time in between which they turn. Yeah. Yeah.
I had a question about the salinity, since you mentioned the sodium concentration. So in freshwater lakes, or maybe in the ocean long ago, geologically long ago, if the salinity was lower, were the bacteria turning more slowly? Do they in lakes? Is that?
It's a good question. I mean, the salinity can actually vary quite a bit, even in the ocean as it is today. If you go from estuaries to areas that are much higher salt content. But I can show you from this picture. It actually takes, so this is ocean concentration, something like 500 millimolar. So you'd actually have to have a huge change in the salinity to actually see it change out in the ocean. Whether cells in freshwater do this, I'm not sure. We haven't explored that yet. But it's a very good question. I don't know what types of cells populate that. And over geologic times, people use bacterial cells because they can evolve actually quite quickly. So they're probably evolving their motor apparatus with these geologic times to change how they swim along with it. That's just speculation, though.
Have any of your students tried to make a motorboat that works on this principle?
Good question. Yeah, this is a principle that you could apply at the macro scale very easily. And we've spoken to some roboticists at MIT that are extremely interested in this idea. So it's the roboticists kind of salivate over these kinds of systems, and it's ridiculously simple. So you could, we've speculated about systems where you could have small robots that they have a propeller of some kind and then a body, and when you want them to change direction, all you do is have them speed up a little bit, and that could create an extra force and buckle the linkage between the propeller and the body. So you can have a very simple system like that can create some interesting dynamics. So yeah, it's a great question, and I think you're probably going to see somebody developing that pretty soon.
Yeah, I just wondered if you could elaborate a little bit more about the motor system, the role of sodium, and is ATP, I assume, is still involved in this. How's that work?
I'm not the person to ask, so we typically don't go inside the cell. No, that's a good question. I don't know the answer to it. I'm sorry.
I hope you're the one to ask this one over here.
Sorry, yeah.
Can you explain your parlor trick with the dye and the corn syrup?
Sure. Yeah. It's very simple, and it's not my trick, but I'll take credit for it if you want to give me credit for it. What happens is if I were to put a dot here, right? Let me define my circle by a couple of dots. If I wind the cylinder around, these little dots, which I can connect or not connect, it doesn't matter, these will just follow circular trajectories that go around and around and around. Right? So if they all do that, if I just reverse the motion of that, they'll follow those trajectories right back to their original position. So it's really just geometry, because there's really no diffusion on these time scales of the dye molecules. If we waited long enough, the dye would diffuse out, and then it wouldn't be reversible. So it's kinematic reversibility, not thermodynamic reversibility. So it's a slightly different principle, but you're absolutely right. You do dissipate energy doing this, but this is purely just geometry, because of the flow lines that these fluid particles or dye particles follow.
So you could put beads there in a line, and then you would see
Yep. Absolutely, yeah. Yeah, if the conditions are just right at low Reynolds number. One thing that is not conserved, though, is if you have something flexible and it bends during this process, that it doesn't come back to where it started. Like if you put a string in there, the string will deform and then not come back to its original shape. So,
were you able to determine that rotation, the speed of the rotation stays constant when these bacteria were changing direction? You can accomplish the same thing by changing the speed of the rotation as far as flexing the shaft.
so the speed does change because it's changing from one direction to the other. So you have a minus twice the speed change in the speed, right? And the speed certainly slows down when we take the sodium out. I guess one of the things that I kind of glossed over was that the propulsion speed is it's directly proportional to the rotation speed of this motor as well. So the slower, if you change the rotation speed by 2, you change the propulsion speed by 2, right? So they're directly linked. Does that answer the question?
I was just thinking that if the cell motor, rather than changing directions, would change its speed.
Oh,
it
right.
would still
Yes.
bend.
Yes. So like my little thought experiment of the little robot that speeds up and buckles the thing.
Yeah.
So so we've done extremely careful measurements of the speed. So this is the speed during this process, right? So green is backing up, so it's about 60, and then it comes to 0 when it reverses direction at this point, and then it comes back up to about the same speed. There's some fluctuations here, but that was actually the first idea that we had when we saw this. We said, oh, maybe it's speeding up just before, to give it a little kick. But yeah, we had to rule that out, and then it made the problem harder. So.
First off, a question: is the buckling similar to like the way my house buckles in an earthquake? Because
in a sense,
it just buckles and it returns, we hope, to its original position. My question is on the sodium influx. Does it work like human muscle does? Where
I don't know how
human muscle, when your muscle contracts, the membrane gets a signal from your brain, and it changes the permeability of the membrane, and you get an influx of potassium across that membrane.
Yeah.
And then if you want to, then you have to push it back against the concentration gradient to make it happen again. And so I would assume that's what the sodium is doing.
So these guys have ion pumps that can push sodium in or out. To that. And
just curious whether it was the same mechanism.
Exactly. Yeah, you're absolutely right. Yeah.
Time for one more question, if anyone is interested. Otherwise, I have a comment about, I don't know the specifics of how this exact motor works either, but the general idea is that these proteins have different stable conformations. They have different shapes. And when the sodium comes through, the interactions change the shape of the proteins in a way that can produce like collective movement. And so that's the only level of detail that I know, unfortunately.
It's better than my level of detail. So.
If there are no other questions, then let's thank the speaker again. So next, I believe we have a town hall discussion. Greg, are you here?